Okay, so yes, let's jump from the 19th century to the 20th, and just a few facts, because of course we'll come back to these things in the next lectures. Computational methods arrived, of course, in the 20th century, and in 1950s people started using mathematical methods to do classification. And that was an important step to bring in mathematical methods into a domain that was so far the domain of taxonomists, and taxonomists were biologists comparing traits, etc., with methods of classifications that were not always rigorous and that were very often prone to criticism. So 1950s, first numerical clustering methods for classification and phylogenetic inference. In 53, first protein sequencing by Sanger, and of course you know that the sequencing techniques have enabled major breakthroughs in the 1990s and then 2000. So first protein sequencing in 53. Same year, Watson and Crick discovered the double helix structure of the DNA. 55, discovery of the DNA polymerase, so a key biological actor of DNA replication, and DNA replication, of course, is key to mitosis and meiosis. 1960s were the first phylogenies from gene frequencies, so phylogenies on traits that were quantitative. Parsimony methods, likelihood methods have been developed in the 1960s, and these methods are still in use today to build phylogenetic trees. And of course, in the 60s, 70s, scientific computers became affordable, and some universities were able to use computers, etc. And of course, since then, it's been ever-increasing computational power available to research scientists. The picture on the left shows Luigi Luca Cavalli-Sforza, who was key to the development of computational evolutionary sciences, and Andrew Edwards, 1963. So I showed the picture of these two guys because I want to talk a little bit about a paper published in 1967 in the American Journal of Human Genetics by Cavalli-Sforza and Edwards. And it was a seminal paper about that was about to to bring to the community of researchers on evolution techniques to build automatically trees from traits, and in this case they used gene frequencies and compared gene frequencies or allele frequencies among various populations to determine how these populations were related to each other, how they diverged from an ancestral population into the extant populations. My dog is drinking, sorry for the noise. So yeah, on the left you see this picture from the paper that shows that we can see a phylogenetic tree as the projection on a plane of a tree whose z-axis would be the time. So you see this is an ancestor, a common ancestor to the different populations A, B, C, and D. And this common ancestor is defined by a frequency of a gene B and a frequency of a gene A, or allele B and allele A. And this ancestor evolved into several subpopulations or several populations. And basically, what we try to draw is the phylogenetic tree linking these different extant populations, and it's interesting to see that the projection on this plane of this three-dimensional tree. So the problem is knowing just the points A, B, C, and D, meaning that each population is obviously can be represented by a point. Once we know the frequency of the allele A and the frequency of the allele B, and the mathematical problem is to infer the evolutionary tree here in space and time. So of course, for that we need to model processes of evolution. So we must make hypotheses about the mode and the speed of evolution, and that's a model of evolution. And we'll see how models of evolutions are designed and how they act on the branches of a tree later on in this series of lectures. And in this paper, the authors went through a comparison between two approaches that are still very widespread nowadays: minimum evolution and maximum likelihood. So just for you to know this, we'll talk about minimum evolution, maximum likelihood later on, not today, but in the following lectures. Okay, let's have a look now at the mechanisms of molecular evolution and speciation. After we've talked about the theory and the evolution of the ideas in this field of evolution, of molecular evolution and phylogenetics. So various mechanisms drive evolution, and I'm going to present some of them in the next slides. The first one is the genetic drift. So genetic drift is linked to the random sampling from the gene pool of a certain generation to create the gene pool of the following generation. And it's like a random walk in the gene pool, basically. The selection is another mechanism. We talked about it earlier. Selection, the term encompasses processes of natural and sexual selection. So natural selection is adaptation to the environment, and sexual selection is preference in mating, bias in mating patterns. Migration is also a mechanism that drives evolution, and geographic migrations of groups of individuals, but also the fact that pollen is transported by bees, and this is a way for plant species to migrate. And so this has always been an essential feature, an essential mechanism driving evolution. And at the level of DNA bases, mutation and recombination, or at the level of chromosomes, are pseudo-random alterations of the genetic material during the creation of an offspring. So For instance, during meiosis, meiosis is the process by which gametes, so sexual haploid cells, are created. During meiosis, these processes of mutation and, more importantly, recombination take place, and that's why even if you have 100 children, no two of them will have the same genetic material. So mutation and recombination ensure genetic diversity of the gene pool, and that's essential also. So you have many different types of mutation or recombinations. All these processes of alteration at the level of chromosomes. You can have nucleotide substitutions, you can have insertions, deletions, duplications, gene translocations, etc., etc. Let's talk about the random drift. The random drift is the process by which, even in the absence of any selection, any natural selection or any sexual selection, even without that, the mere fact that you have a population and that goes to the next generation, the third generation, etc., etc., with the constant size of the population, the fact that not every individual will have, or not every couple will have as many offspring than the others, make it that there is a random. So the process of going from one generation to the next involves random sampling into a certain gene pool. So, for instance, in these sketches, we represent, for instance, let's say it was about, we just represent one gene, and this gene has 3 possible alleles, blue, red, and yellow, and this is the gene pool for one generation. And at the next generation, because within this gene pool random mating has occurred, with maintaining a constant population size, there is a certain choice that has been made in this population to get to that one. Okay, there is a random. Let's say you have this urn, and you have to let's say take balls, pick these beads with replacement. So pick n beads with replacement, so that at the end you have the same number of beads in the next jar, and you can pick at random. So you may have this scenario, but we can imagine another scenario where at the 5th generation we would have 30% blue, 30% red, 30% yellow. We may imagine a scenario also where in the last generation, in the 5th generation, you have only reds and yellow. Okay, so all these different scenarios are possible, and this is due to the fact that there is randomness in the selection of the gene pool to form the next generation from the previous generation, and this randomness is called random genetic drift.